This video focuses on carbohydrates, one of the 4 organic molecules we'll be discussing. So let's start with a little background information, okay? Carbohydrates are made up of smaller parts called monomers. And a monomer is really just a subunit or a building block of a larger molecule. So the smaller monomer that makes up carbohydrates is called a monosaccharide. Each organic compound has its own specific type of monomer. So you can take a single unit, a monosaccharide, and you can combine them together using dehydration synthesis, which you should have learned about in chemistry, to form covalent bonds between these different monomers. So these covalent bonds are formed by removing water. And now, instead of having a single monomer, we have a larger, more complex structure called a polymer. And in this case, our polymers are going to be our larger macromolecules. So let's talk specifically about carbohydrates. Carbohydrates have 3 primary elements: carbon, hydrogen, and oxygen. And their ratio, their empirical formula is 1 carbon to 2 hydrogen to 1 oxygen. Right? For example, glucose is C6H12O6. Now, this is actually the molecular structure of glucose right here. And this is also an example of a monomer, or a single subunit, called a monosaccharide. Mono means one, saccharides means sugar. So examples of monosaccharides can include 6 carbon sugars, like this molecule. And I'm going to write 6C for carbon sugars. An example of those types of monosaccharides are glucose, galactose, and fructose, among many others. Other monosaccharides sometimes are only made up of 5 carbons. So 5 carbon sugar examples include deoxyribose and ribose sugar, which are the sugars found in DNA and RNA specifically. So here's the confusing thing, right? When you look, let me focus this in a little bit more, make it brighter. When you look at this molecule, you actually don't see 6 carbons. And that's because organic chemists can be lazy sometimes, and they don't like to draw in all the carbons. So anytime there's an intersecting point and no element is specifically listed, that's going to be a carbon. So we have a carbon here, that's listed. This intersecting point will be a carbon, 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 carbon. When you count carbons, you always start at the part of the molecule that's a little different and count clockwise. So this is carbon 1, carbon 2, 3, 4, 5, 6. And all of a sudden, you have the 6 carbons we were looking for. Let's do the oxygens next, because those are a little bit easier to see as well. So let's look at the oxygens. We have 1, 2, 3, 4, 5, 6. So we have the required number of oxygens. Then we come to our hydrogens. Those are a little bit stickier. Right now I have 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. So we're missing some. That's because anytime there's a carbon can form 4 bonds. Okay, so any place there's a carbon and you don't see 4 bonds shown, the missing atom there is a hydrogen. So this carbon is attached to 1, 2, 3, 4 things. That one's done. This carbon is attached to 1, 2, 3 things. So this has to be a hydrogen. This carbon is attached to 1, 2, 3 things. So we're missing a hydrogen here. This carbon is attached to 1, 2, 3 things. So we're missing a hydrogen here. And finally, this carbon is attached to 4 things. This carbon, 2 hydrogens, and an oxygen. So it's set. But this one is attached to 1, 2, 3 things, hydrogen. So now we should have our 12 hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so that's our molecular structure of glucose there. And this structure can change around, but remains C6H12O6. And when it changes where things are located, that's how it can become glucose, galactose, and fructose. Outside of water, these monosaccharides are straight chains. But as soon as glucose, galactose, fructose, deoxyribose, and ribose get into an aqueous or water-based solution, they form rings. So I'll typically show these sugar in ring form, the circular form, because the chemical reactions that we're really talking about happen in water for organic compounds in our bodies. So examples of monosaccharides we listed, uses, right? Glucose, galactose, and fructose are good energy molecules, and deoxyribose and ribose are good structure molecules that form the backbone of our DNA and RNA. So what about if we want to stick 2 of these monosaccharides together? Well, to stick 2 of these monosaccharides together, we can do a dehydration synthesis, okay, which you know from chemistry is also called a condensation reaction. And I'm going to write RXN for reaction. In a condensation reaction, you remove water to form a bond. So I want to give you an example here. In this chemical reaction, the arrow denotes turning into. So these 2 are the reactants, which turn into this singular product, right? So this is a monosaccharide and this is a monosaccharide. So we have 2 monosaccharides. We want to take these monosaccharides and we want to join them together to produce a singular disaccharide. Remember, mono means 1, so this is 1 sugar, 1 sugar. Disaccharide, we have 2 sugars put together to form a disaccharide. Examples of disaccharides include sucrose, lactose, and many, many others. But how does this covalent bond form here? Basically, what happens is we have to remove a water. So what happens is the hydrogen from this sugar and the OH or hydroxyl group from this sugar are removed from these 2 molecules, and they combine to form H2O or water. Okay, so we're dehydrating these molecules. Because these were removed, we have enough energy to form this new covalent bond between these 2 sugars. Okay, so that's how you form the disaccharide. So if you're building disaccharides in the body, that's how you make them. Let's say, however, that you want to break disaccharides apart. Just give yourself a second to remember that type of chemical reaction that you also learned about in chemistry. That's called a hydrolysis. You're going to add water to break bonds. Oops, you can't see that. I'm so sorry. Now you should be able to see it. Add water to break bonds. So for example, if you have a disaccharide, I'm not drawing all the elements in. You could add water in here and then break that bond. So now you have 2 monosaccharides. So when you're digesting your food, this is what's happening. Okay, so now we have 2 monosaccharides. So this process is reversible, but it does usually involve 2 different enzymes. So we have monosaccharides, single, disaccharides, 2 sugars together, and sometimes we can get more complex, much larger molecules called polysaccharides. Polysaccharides are large polymers, and these are a great example of a huge theme in biology, which is that form or structure of organisms, molecules, cells, whatever we're talking about, often inform the function. So structure informs function. The structure of this pen its length, the fact that it's ink informs its function, being able to grip it to write, right? The structure of these scissors right here, the sharp blades, the ability to open and close, informs their function, which is being able to cut things. So let's look at these structures. We'll start with plant polysaccharides first. These polysaccharides are found in plants. Okay, so we have two we're going to talk about. This first one is starch, and you can see starch has many, many monosaccharides. Each of these is a monosaccharide grouped together, but it branches a lot. Okay, so starch is a very branched polysaccharide found in plants. Okay, so form is branching. Oh, I spelled branching wrong. Sorry. Form is branching, right? Which leads to more surface area to increase the rate of breakdown by enzymes, and which leads to its function, which is to store energy. Because basically, because it's very branching, the enzymes can get here and break these apart and easily release energy or put them back together to easily store energy. You can see with cellulose, cellulose has the same monosaccharides, but it is all these monosaccharides are stacked on top of each other and connected. Okay, so the form is straight chains with hydrogen bonds. Because of the structure, we've increased stability. And because of this, the function of cellulose in plants is structural support. It makes up cell walls. So interestingly, we eat a lot of plant starches and we use these for energy. We eat a lot of plants that will have cellulose, and our bodies don't have enzymes to digest these well, but we use this as fiber. It helps scrape out your intestines so that you can basically poop more easily. So structure determines function, and they're very different. So you need to remember these two polysaccharides are found in plants. Next, we have animal polysaccharides. And these aren't the only polysaccharides that exist. These are just the ones I'm talking about today. First, we have glycogen. If you look at glycogen and compare it to starch and cellulose, you can see glycogen looks a lot more like starch than cellulose. So if you had to make a hypothesis about its function, right, it's very branched, that's its form, you would probably think that it stored energy if you made that hypothesis, and you'd be correct. So form is very branched, which we know increases surface area, and function is energy storage. It's even more branched than starch, so it can store even more energy, which makes sense because animals have higher energy expenditure as compared to plants. Usually you find glycogen in storage in your liver. Chitin, okay. Chitin looks a lot more like cellulose, not exactly, but a lot more like cellulose. And chitin is more stackable, and it makes up exoskeletons in insects. So again, it's structural. And it also makes up fungal cell walls. So this isn't an animal, but same thing, right? This is very structural. So chitin is more similar in shape to cellulose. So this is the end of my video on carbohydrates. Thanks for watching.